Good morning, students. In this video, I shall discuss the phenomenon of linkage and recombinations. Now, what is linkage? This process of linkage, it is normally observed by Batson and Ponnet in 1906. And according to him, actually earlier to Batson and Ponnet, normally as in case of Mendel, he gave the law of independent assortment. He gave the law of independent assortment. And according to his independent assortment, normally the genes are segregated or separated from one another independently from during gamete formation. But this law of independent assortment, it is only true when the genes are located in separate chromosomes. And normally when Mendel he selected the pea plant as his experimental tool or experimental material, and that is why he surprisingly, or you can say it is his advantage that all the genes he taken into consideration, all the genes which he taken into consideration for his experiment are present in separate chromosomes. So that is why the genes segregated from one another independently and in the gamete formation or during the formation of the new organism of the next generation. But what happens if the genes are not present in the same chromosome? Then only it is true. But if the genes are present in the same chromosome and according to the chromosomal basis of inheritance, if the chromosomes are separating, not the genes, then what happens? The genes which are present in the same chromosome, normally they tend to segregate together. They never separate from one another normally. And during gamete formation, all the genes which are present there in the same chromosome, suppose this is a chromosome, and whatever genes which are present here, these genes, they normally segregate together or transmit to the next generation together. They do not separate from one another. This phenomenon of transmission of these genes together from one generation to the next generation or from the parents to the formation of the gamete then to the next offspring. So this phenomenon is called linkage. And this linkage was observed for the first time by Batson and Ponnet in his famous tool that is called Lathyrus odoratus, which is also called a sweet pea plant. So here what happens? The genes which are present in the same chromosome, because the genes are responsible for the expression of the characters. So if the genes are present in one chromosome, means all the characters related to the gene, they will be there in the next generation as their parental combination, and the same combination which were there in the parent, this combination will be observed in the offspring. So the parental combinations are normally observed in offsprings. So that is why no variation, no change is normally observed in the character of the offspring. So this phenomenon is commonly observed there in the linkage phenomenon. Now, how this linkage group or this group is formed there in a particular chromosome? So according to this, all the genes which are present, they are in a particular chromosome. They normally transmit from one generation to the next generation together. So that is why the genes which are normally present in the same chromosome are called or are considered to be one linkage group. So this linkage group, which is normally according to the number of chromosomes or number of haploid sets of chromosomes present there. So what is a linkage group? So this linkage group means the genes which are present, the genes present in the same chromosome, it is called a linkage group. So if in case of human being, there are 23 pairs of chromosomes. That means there are 23 haploid chromosomes. So in this case, what happens? In case of human males, all the haploid chromosomes of up to 1 to 22, there are 22 chromosomes. And now the allosomes are two different types in case of males. So that is one X type of chromosome and another Y type of chromosome. So that is why 22 types of chromosomes there, 1 to 22, then X and Y. That means there are 24 types of linkage groups you will find there in case of human male. But in case of human female, there are the autosomes are same 1 to 22, but the allosomes are of one type, only X type of chromosome in the female. So that is why only one type, that is why there are 23 types of linkage groups is found there. In case of types of linkage groups are found here in case of human female. Now in case of pea plant, there are 7 chromosomes, that is why there are 7 chromosomes. So that is why there are 7 linkage groups are there. So in case of male, there are 11 chromosomes. So that is why there are 11 linkage groups are there. So here 11 linkage groups are there. So these linkage groups are normally corresponds to the number of haploid set of chromosomes in that particular organism. But this linkage phenomenon, it is normally observed always in all the organisms where the genes are present in the same chromosome. Now, earlier Mendel, luckily he found the genes which were observed in the different chromosomes. That is why this linkage group was not observed there. But here the linkage groups are normally there which are present in the same chromosome. Now the next important thing is that whether always from one generation to the generation the same genes remain together or occasionally or sometimes they normally separated from one another. Now it was also observed that sometimes these genes are separated from one another, and this phenomenon is called recombination. Now the recombination. This recombination process is directly related to the linkage phenomenon. Linkage means the transmission of character from one generation to the another generation together. That means the genes which were present there in the same chromosome, they are inherited to the next generation in the same combination. That means the same parental combinations are observed in the offspring. But normally, sometimes what happens? Some new combinations are observed in the offspring. Now this process of reshuffling of the parental genes which were remain together. Suppose their genes, suppose this is a chromosome and these are the different genes which are present there in this particular chromosome. But in the offspring, suppose it is the parental combination, and in the offspring, suppose these combinations are different. Then what will happen? Now why this difference occur? So this reshuffling or the difference in the gene combination in comparison to the parental genes. Okay, then this production is called the recombination. So this reshuffling of parental genes to produce a new genotypes. Normally, whatever genotype present in the parent, the parental, it is called the parental genotypes. But when there is reshuffling occurs, due to reshuffling, some new genotypes, some new combination of the genes were present in the offspring, and these new recombinations or new genotypes of the gene is called the recombination. That means whatever combination was there in the parent, it is actually changed or it is reshuffled, and a new combination of genes are observed in the offspring. And that is why in case of offspring, the characters are different from their parents. Now, why does it occur? It occurs normally due to the crossing over phenomenon which occurs during meiosis. Then what is meiosis and how this crossing over occurs? It will be actually as already discussed in class 11. Now in cell division chapter. Now during meiosis, what happened? The pairing of the homologous chromosome occurs, and after the pairing of the homologous chromosomes, now exchange of the segments between the non-sister chromatids of homologous chromosome, non-sister chromatids of homologous chromosomes. So when the homologous chromosomes paired together during meiosis 1, what happened? The non-sister chromatids of this homologous chromosome, they exchange their segment, and this exchange of segment between the non-sister chromatid, it is called crossing over, and normally the chiasma formation occurs. So the crossing over and chiasma formation, this process occurs there, and due to this process, the random fusion of the gametes also occurs there. So due to this crossing over phenomenon, that means exchange of segments between the non-sister chromatid, that means exchange of the genes between the non-sister chromatid, and the random fusion of these gametes. So due to which this type of recombination occur, this type of variations occur, and this type of new type of combination, that is why observed here. Now, according to Morgan and Sturtevant, normally they observe that how the number of recombinations actually occurs there, and how it can be predicted or calculated. According to Morgan's and Sturtevant's hypothesis, the frequency of crossing over or frequency of recombination of the genes is directly proportional to the distance between the genes. That means if the distance between the genes is more, then automatically the rate of crossing or number of crossing over or frequency of crossing over will be more. So two genes are suppose here wide apart from one another, the chance of crossing over is more, and if the two genes are normally closely present one another, then the chance of crossing over is less. That is why the chance of crossing over between the genes which are present in the same chromosome is more when the distance between the genes is more. So that is why the frequency of crossing over is directly proportional to the distance between the genes. And if the distance is very less, then there is chance of crossing over is also less. And that is why this recombinant frequency is normally directly related to the distance between the two genes. Now, this recombination frequency, or in short it is called the RF, it is equal to the number of recombinations, so how many recombinations occur there in a particular chromosome, and divided by number of progeny, how many number of progenies are formed and how many number of recombinations are there actually formed. So these two data, it actually indicates the recombination frequency for that particular organism or particular species. Now, the maximum recombination frequency between the two genes is 50%. That means there is 50% chance maximum is there for the recombination process. Now, 0.01 recombination frequency, it is also called the 1% recombination, and 1% recombination, it is also taken as 1 MAP unit, and this unit is taken as centimorgan according to the name of Morgan. Now, 1 MAP unit, it is the number that actually corresponds to the recombination frequency of 1%. If there is a recombination frequency of 1%, then this is called 1 MAP unit. Now, so that is why this recombination phenomenon, it is commonly observed in almost every species, every organism. And that is why their offsprings or the characters which are present there in the offspring are not exactly same to their parents, because during the crossing over, it is the process of sexual reproduction. During meiosis, during gamete formation, what happens? When the gamete formation occurs, at that time the meiosis occurs, and when meiosis occurs, at that time what happens? The crossing over occurs, and due to crossing over, exchange of genes occurs, and due to exchange, the new recombinations are formed. Now, this process is also explained by Morgan in his dihybrid cross experiment conducted there. In the dihybrid cross experiment normally conducted in Drosophila. So in Drosophila, he conducted the experiment, did the dihybrid test cross experiment, and in his dihybrid test cross experiment, he actually explained the process of recombination, how it occurs in those organisms in a well-defined way. Now, this video must be useful for you, and if it is really useful for you, then please like it and subscribe for further videos. Thank you.